Welcome to this video on the single gene survey, where we will explore the marker gene approach in metagenomic analysis. We begin by defining single gene surveys, which are a focused method within metagenomics. Marker genes are conserved genetic loci that act as taxonomic barcodes. They allow us to classify entire microbial communities based on sequence variants within these specific regions. The 16S rRNA gene is the gold standard for bacterial phylogeny. Its key features include universal conservation, 9 hypervariable regions, and sufficient length for phylogenetic analysis. Single-gene surveys are beneficial due to their cost-effectiveness compared to shotgun sequencing, the existence of rich databases like Silva, and their high-throughput capability for processing many samples simultaneously. The first step is sample collection. This involves careful retrieval of samples like soil or water, along with immediate stabilization or freezing to prevent changes in microbial composition. Next is DNA extraction. Successful isolation of total DNA requires breaking the cell walls of diverse species to ensure unbiased representation of the community. The third step is PCR amplification using universal primers. These primers target the conserved ends of the gene, flanking the hypervariable regions to specifically select the 16S gene. PCR primer dynamics involve achieving universality to target a broad range of taxa while managing primer bias, which can distort community representation. Optimization balances broad reach with specificity. Sequencing, using next-generation sequencing, allows for the parallel sequencing of millions of fragments. This depth is essential for detecting even the rarest members of the microbial community. The bioinformatics pipeline includes denoising to remove sequencing errors and clustering. Clustering groups sequences into ASVs, or amplicon sequence variants. Microbial units can be defined as operational taxonomic units, or OTUs, clustered at a fixed similarity threshold. They can also be defined as ASVs, which are high-resolution variants distinguishing single nucleotide differences. Let's now consider how to understand genetic relationships in the context of single-gene surveys. Orthologs are genes in different species derived from a common ancestor via speciation. Paralogs are genes within a genome related by duplication events. Marker gene analysis infers evolutionary closeness by comparing sequence identity. Finding orthologous sequences allows us to map phylogenetic relationships within the microbial community and its environmental niche. The taxonomic profiling comparison highlights the relative abundance of key phyla, like firmicutes, Bacteroidetes, and Proteobacteria in the sample, showing their distribution within the community. Marker gene studies offer high cost efficiency, processing many samples for a fraction of the cost of shotgun metagenomics. This makes them excellent for large-scale biodiversity mapping. Current limitations include difficulty differentiating at the species or strain level, being blind to function, and the issue of primer bias, where some groups may be consistently underrepresented. The future of marker gene analysis involves hybrid pipelines, integrating 16S data with metabolic modeling, and AI pipelines for automated error correction to improve accuracy.